Hello everyone, welcome to the Reproductive Technologies station of Stem Cell Talks. My name is Jacob and today I'm going to be talking to you about assisted reproductive technologies and how we use them in our lab. Now, in vitro reproductive technologies or assisted reproductive technologies essentially utilize gametes or embryos in vitro or in glass, and that's a lot of what we do in the lab is develop embryos. But before we get started talking about embryos, how do we develop embryos? Well, as you may know, we use the male and female complement of gametes, namely oocytes and sperm. Now, for oocytes, we get them from ovaries. Unfortunately, I don't have ovaries with me right now. Usually we get them from a slaughterhouse, but this is kind of what they look like. And essentially within this ovary, we have different blue circles here, and those are called follicles. And the follicles are what house oocytes. And so we use a needle and using a vacuum just aspirate those follicles and get the oocytes. Now for sperm, it's really cool because in our lab, we have a bunch of cryopreserved sperm, millions and millions of doses of bull semen here. And what we can do is, here's an example. What we do is we have some straws here within these canisters, and all we do is we thaw one of these straws within some water. I'll just do one right now. Put that there, and we'll let that thaw, and I'll show you guys what the end product looks like. And while that's thawing, I'll just talk to you a little bit about fertilization. So after fertilization, when the gametes meet, they form a totipotent stem cell called a zygote. Something like this. As you can see, it's a single cell, and it's a totipotent stem cell. Totipotent means it can derive into any different cell type of the body, whether it be a skin cell, a heart cell, a liver cell, etc. Now, what we can do, I'll just show you what we can do with the sperm here. I'll cut this guy open. What we do is we cut the top of this straw like this. We flip this over. And if we cut this side, it's kind of like a vacuum. It'll release the contents of that sperm into this Eppendorf tube. And this is the actual sperm that we use in the lab to fertilize our oocytes to generate something like a zygote. Now, after in vitro culture, which can take up to 6 days in humans, and we can culture these zygotes into blastocysts in things like plastic bags, which is what I do. You can actually see the dish inside there. Or you can use something like a portable incubator, something like this, in addition to a regular tissue culture incubator. And so after about a couple days, you'll see cell divisions called cleavage. Within reproduction, we call it cleavage, which is cell division without an increase in size. And so you can see a 4-cell embryo here, and it's relatively the same size as that single-celled zygote that we talked about earlier. Now, these 4-cell embryos are still totipotent, so they can still become every single cell within the body. However, at the point of the blastocyst formation, about day 6 in humans, we get what's called the first differentiation event, where you have an inner cell mass here, this thicker region that you can see, that actually becomes the developing fetus, develops into the actual organism, whereas the surrounding cells here become what is known as the placenta. And so these surrounding cells are called the trophectoderm or trophoblast cells, and so they don't actually contribute to the organism. And so once we have these blastocysts, what's really cool is we can take this inner cell mass section, this thick ball of cells here, and we can derive it into an embryonic stem cell line, something like this behind me. And so these embryonic stem cell lines, they're really cool because you can immortalize them. And these still remain pluripotent, so they can become any cell type of the body except for placental tissues. And so these can become immortalized. There are actually 2 embryonic stem cell lines in Canada for humans. One's actually at the University of Calgary, which is pretty cool. Alternatively, if you don't want to turn your blastocyst into an embryonic stem cell line, what we can do is we can freeze or cryopreserve them in something like this. This is our embryo freezer, and we can freeze them, and whatever later date we want, we can then implant that blastocyst into the uterus of an animal, and hopefully a new life is born. So yeah, with that, that's everything you need to know about reproductive technologies and how it relates to stem cells. So thank you so much, guys. I hope you're interested and I hope you enjoyed.